The topic of my presentation topic is the CSS. CSS, the full meaning of CSS is the composite synergistic system model. It's a dynamical model for the selection of the individual patient-specific cancer therapy. Uh, Mostly AI or that sort of work or any statistical method actually identify a group of people, but here these models are specifically targeted for the individual patient. So this this thing, you know, that any cancer actually survived by the process of angiogenesis. So how angiogenesis occur? From the nearby blood vessels, some protrusions occur, or these are called microvessels. These microvessels actually supply uh, the tumor, and tumor actually survive based on by getting nutrients and oxygen through these microvessels. So conventionally, uh, all the cancer patients are treated with the maximum tolerable dose, and that has some, definitely some has disadvantage, that it makes a high toxicity and immune suppression. Moreover, the maximum tolerable dose causes delay in therapy. Why? Because due to the high dose of chemotherapy, these microvessels rupture, but the drug will reach to the tumor site through these microvessels, and MTD is actually uh, targeted to the cancer cells directly. So some other form of therapy is suggested, that is maximum tolerable dose with the intermittent hematopoietic stem cell transplantation. That hematopoietic stem cell transplantation actually helps in development of these microvessels. And another form of therapy is that is called metronomic chemotherapy, which is a dose dense therapy. That means very low dose but frequent interval. The same drug is applied, or the same chemotherapy drug can be applied in the MCT chemotherapy mode, where the drug actually target the microvessels instead of targeting the tumor. So the tumor actually destroyed by lack of nutrition. So I have already told that this is a composite synergistic systems model. It is basically a combination of two models. Uh, One is the vasculature growth model, and another model is the fluid dynamical model. In the vasculature growth model, where number of the vasculature cells are being considered, where in the FD model, the vasculature diameter was considered, and but the both actually targets to the tumor angiogenesis. The objective of this such sort of work is to check the drug application strategy, the timing of drug application of the drug, the dose, and the long-term therapeutic benefit. And this is basically a system designed for the anti-angiogenic drug. Another drug, another drug is also suggested for the last 1.5 decades. Uh, that is mostly they are the antibody-based drug, and for the actually the actually the uh, previous model was developed with the VG model, and that is with the anti-angiogenic drug. Next is the next fluid dynamical based model, actually based on with the MCT strategy and MTD strategy. And I already told that the development of the microvessels process, development of the microvessel cells or microvessels, and how the MCT works. So far, what we know about the mechanism of MCT action is that the MCT acts on the extracellular matrix, and to release the a molecule TSP1 that bind with the CD36 of the endothelial cells that actually inhibit this binding of the drug with this molecule CD36 causes suppression of the endothelial cell growth. Moreover, TSP1 also sequestered the VEGF, uh, vascular endothelial growth factor, from the tumor cells. So the microvessel, if the microvessel cells has not been developed, then tumor growth development is not possible. Now, for the VG-based model, vasculature growth model, three main factors were considered. One is the drug sensitive, drug resistive, and vasculature cells. Now, an anti-angiogenic drug is applied on these VV cells, whereas full FD model is based on the two same thing: drug sensitive, drug resistive, vasculature cell diameter, and another factor was considered, that is TAF, tumor angiogenic factor. It is further being considered with by making a 3x3 grid system where the probabilistic movement of the this vasculature is being considered, because numerous vasculature, miniature vasculatures are being formed at the tumor site, and here the FD model drug is applied in this manner. The black arrows and the MPD is applied to the blue arrows. Now, both the models has some pros and cons. Why actually there is need for the development of the CSS model? Because individually both models has some advantage and disadvantage. Like VG growth model, for any mathematical model or analytical model, one has to initialize the system. So for the initializing system, cell count is directly can be added for the VG model. But in the FD model, the initial the MBD or MBD, the number of MBD cannot be introduced very directly. The disadvantage of that VG model is that the intermittent tracking or follow-up, making a follow-up to biopsy is very difficult. Whereas FD model has advantage that it can be follow up by through MRI or CT scan. The disadvantage is that though we know that the very two consecutive, if MRI or CT scan is performed between two consecutive very close time, then it may not produce any appreciable data change. So all these things are being make together to make the CSS model. And another important thing is that in the FD model, TAF was considered, that TAF is a soluble factor that can be tracked throughout the follow-up. So this is the this is the just hold on. This is the overall make the synergism in the system. And here another important thing is that MCT, small dose of chemotherapeutic drug, can stimulate the immune system, and that has also been considered or introduced in both the models. And another is from the bone marrow, hematopoietic stem cell mobilization is also being considered in the CSS model. This is the representation of the overall VG model, and this is the FD models. And another thing is that the tracking, I told that the TAF is being considered, and TRAF has been introduced in the model, where in the grid system 2.2 position is being considered and that position was correspond with a number of cells in the tumor, and the distance and the TAF is TAF is being calculated through the number of tumor cells. So this is the another further we make the actually the both the models, the CSS, in fact the VGFD, all are actually nonlinear model. So in the nonlinear model system, we cannot identify the control theory, because control theory is very useful uh, or maybe very effective for identify or pinpointing the position a priori state, to identify the at which position the tumor become unbounded. So we make an approximated model from both the from both the variables, from both the models, for the control analysis, and it is a linearized model. Now we make a simulation study, and here uh, both the models are synergized for the free growth condition, so that we can assure that the both models are start from the same position, and we consider it's a vigorous type of tumor growth. And for that, we put the data from the known literature for vigorous type of tumor cells, and and the same tumor condition is being studied or being studied through simulation analysis for different sort of therapy, that is MTD, MTD with intermittent HSC, AAG, MTD with HSC followed by AAG, and MCT with immune boosting. And these are the output results that we have found. And here we have seen that the MCT is much far better with obviously with immune stimulation. And these are the simulation data that we have found, that these are the drug strategies. The blue left panel of the blue line is a drug strategy. This is red is the intermittent HSC transplantation. These are the how the number of the doubling time of the, I have already told that the change of the tumor condition is also being happening. So we put that system into our modeling approach, and this is the TAF dynamics. You see that this TAF is gradually being increased, and it's reach a plateau site, and this shows that the only MCT there is a gradual decrease, and it's become negligible. It is all the simulations are being carried out with the same, considering the same tumor condition, and this is this is the in detail the TAF position of at the different grid points. And we have also from the tumor dynamics, it is also seen. We have also make relationship of the tumor tumor radius, because tumor radius can be easily be observed through MRI or CT scan. So here also shows that MCT is quite effective. So this is the summary of the whole simulation study. Now the uniqueness of the approximated system is that the original system and the approximated system, the original system in the free growth condition, that means when no drug is being applied, it comes earlier, whereas approximated system followed the original system. So this whereas in the for the individual cells, it actually correspond to the both correspond on the same line. So, but in this case, original preceded precede before it, whereas the approximate is a later it. Uh, it's a sort of limitation, because a priori prediction is made, but you see that up to initial state, up to 250 days, this is x-axis is a day, and this is a tumor volume. Up to 250 days, it is preceded the actual system. But in the growing condition, when the tumor has full grown, then it's expected that it cannot predict very perfectly. But we are basically make this approximated system to make the a priori prediction. So in that sense, it is successful. Now, the data for both the models need across the scales. So already some suggested procedures are there to obtain those data, like TAF. TAF can be monitored by ELISA or mass spectrometry. RAS or APC can be followed by the biopsy or real-time PCR. Determination of the cell doubling time by the ex vivo cell culture. Tumor cell cycle can be done by the flow cytometry. Immune activity can be cell immune activity or ELISA for inflammatory cytokines. These are the or the tumor volume or blood vasculature can be made by CO, MRI, or immunohistochemistry of biopsy samples. So these way the data can be fit to the model. So I already told that this is our overall observation of the simulation results. That is, MTD HSC age is better than MTD HSC, and MCT is much better than age. So I already told that MCT is much better in our simulation result. And I already told all these things, that comparative basis. So now the importance of the CSS model, I already some discussed, that the a priori prediction of the future state, long-term therapeutic outcome, and the most important thing that it can be made for the individual specific therapy design, drug optimization. We can make, depending on the initial tumor state, we can make the drug make the drug dose, and when to apply the drug. So that can be made, and it can be changed along during the course of the therapy as well. And we hope that this model has definitely has some implication for the cancer systems medicine. And in the drug repurposing, the concept is that not to change the drug, but the same drug can be used in a different manner. So this model can help in the drug repurposing strategy as well. So these are our publications, and we are actually working on different in the field of systems biology and systems medicine, and as well as systems pharmacology. So we made some made a society, scientific society, so anyone can interested can go through this, check our activities over here, and this is our blog. One can write in this blog. This is open to all. So I invite all of you to put your opinion and comments, as well as if you want to make any further contact in future, you can go and visit that our site. Thank you all.